Thank you so much for carving time, an extra day out of your very busy schedule to join us here for our second ALS Familial Summit. Nearly 600 people have joined us over the past 3 or 4 days going to the Hill in an effort to increase the awareness of ALS and to continue to ask for support in our search for a cure. Jane Gilbert, our President and CEO, really needs no introduction. I know many of you know her personally, as well as our next speaker, Dr. Lucy Bruin, whom, again, probably needs no introduction because I know many of you have spoken with her directly and personally. Dr. Bruin is our Chief Research Scientist with the ALS Association. She is an international traveler and an international expert in the ALS community. She's going to be sharing information this morning with us. So without further ado, join me with a warm welcome for Dr. Bruin.
So yes, you're stuck with me again. I'm sorry for those who heard me yesterday, but I hope to tell you something new. We were trying to juggle it actually that Brian would be presenting, Dr. Traynor would present first, because really his discovery in the C9, it's really his discovery, so I don't feel that I would be the one to be talking about it. He hasn't quite come yet, we hope he's on his way. But what I will do, and so I know you heard quite a bit yesterday from me, but what I will do, rather than giving the whole story of C9, I will give you some of the updates of work we're doing. So you'll get it a little bit in reverse, but I think you'll understand the field anyway the way I presented, and then you'll hear the story in person from Brian, because really it's his finding. So let's fire away. Sorry, I've just got to see. This is I always have to move around, so I feel a bit stuck here, but never mind. Okay, so, we all use this image because it's really the excitement in the field, and I think it's a great success story, and you saw it yesterday as well. For all of you sitting in the room, you clearly have a connection, a loved one, or yourselves have familial ALS. And in part, that's very scary, and I can completely understand it. And it's very difficult for me to say here today, gosh, but you really are the ones that we know the targets and we find the therapies. It's a slow process. But in reality, my biggest hope for treating this disease effectively is through the strategies that we are going to use now, is directly to attack the target. For those with sporadic disease, I am also very hopeful, and in fact, we learn the lessons from the familial cases to be able to bring it forward into sporadic disease. I know sometimes I get questions and emails from families who do not have a family link, who feel frustrated that most of our focus, we seem to talk about familial ALS, the familial genes. Well, the field is shifting so importantly because actually the genes we find in families are very often also found in sporadic cases. It does give the interesting story, and I know Dr. Traynor will tell this very well, is that can we be sure if you carry a gene that it will go on to the next family? And there are some newer genes, the C9 in particular, where there's a lot of mystery around that. We don't know for sure. And just actually on the Hill, the story of a family where there was a younger woman who is living with the disease now, and at the same time that she was going through the diagnosis, there you go, your mother passed away from it. That to me is an incredibly puzzling tale, because we can't figure that out. So, there's a lot more to it, and I do believe that our lessons from the familial cases and the familial people with ALS will help us in understanding treatment for everyone. But this is so encouraging, and Dr. Traynor will tell you that there's a confidence that that pie will be filled within the next few years. I don't think that chunk is going to be as big as that C9 family. This is a remarkable finding, and it was a very challenging one, and I do love that story. And for me, from a funding perspective, I'm sometimes challenged. In fact, yesterday was very challenged in our research committee meeting. Why are we funding so many in a similar area? And actually, if we hadn't funded 3 different investigations, and with me being able to connect with all these people behind the scenes and respecting confidentiality while we don't have the answers, we may not have got there as fast. It seemed slow enough. It was 7 to 8 years. But there was a need to use novel technologies to build what we now use for the mouse models. But there was also a need to go back to some very different techniques that actually I'm so proud it was 2 young investigators who are up and coming rather than the seasoned established investigators. So it's really been tremendous for the field and important for us to always consider funding multiple activities because we actually ultimately want to speed this up. So that's a long story to tell you that we have now several genes. And what do they do for us? They help us build model systems. And in 1993, when I joined Don's lab, about to launch into Alzheimer's research, the discovery of the SOD1 gene suggested to his team that actually maybe we will change direction. I came as a specialist in Alzheimer's disease and was working on a molecule that Don Cleveland was very passionate about. Aha, we have Brian. Good, sorry, just glad that you are here. So just to fill you in, Dr. Trainer, I'm going to do nothing about telling the long story of C9 because I wanted you to introduce that and we're just reversing it around. But I'm here now, so I'll take the stage and finish off. But really, the exciting thing about that mutation was it really marked the beginning that scientists had tools to do the research. And it's because of those tools that we have so many people dedicated to ALS research. We can build mouse models, we can test ideas in yeast, which might be, seem very strange because it's so far away from a human system, but we've learned a lot about processes in all these different models, flies, worms, zebrafish. A wonderful story that Dr. Sokovitz told yesterday of a finding in the zebrafish, which was a gene that they picked up through a screen in this little fish that sits around in a tank. That was interesting. They then went to humans and found that that particular gene had changes in it. And in fact, if you had a gene that carried those changes, your disease was slower. So it is remarkable what we can find from model systems as long as we always connect it back into human systems. And at many, many podia where I stand and talk about the value of these systems, there is a lot of challenges. While that mouse model, we've tested everything in it, and everything has failed. We all have different opinions. Frankly, I don't think we've tested anything in terms of a small molecule that has given any significant impact in the mouse model. 2 to 3 weeks is not really a big deal. And so I just think we haven't had the blockbuster drug in that mouse model yet, my opinion. But I do also think it's very important that we use many different interim steps to tease out whether that mechanism that we're trying to treat is actually important in the human disease, or is it one of the many mechanisms and it's insufficient to correct that to get a big effect. But model systems have been phenomenal for us. I also highlight this because she's been a researcher very, very long committed in this area of looking at the upper motor neurons. And yesterday I told you, to many of you, that the whole system is, in ALS, it's the upper and the lower motor neurons. And the SOD mouse model has been very useful in that we've focused a lot on the lower motor system and the connections. But in fact, tracking very carefully and building tools, there's now ways to carefully look at what is happening with the upper motor neuron system. And just in our funding decisions last night, we're about to fund an investigator who is going to actually make iPS stem cells, so induced pluripotent stem cells, drive them to a phenotype of upper motor neurons, and that's the first study to do that kind of thing. So really exciting that we are on the cutting edge of building tools and building techniques. I did mention yesterday as well, and I think this is interesting, especially in light of discussions with the limitations of model systems and the mouse being so far away from the human, the fact that there are dogs that carry the SOD mutation. And in fact, Joan Coates, who is a veterinarian, is really focused on this study and working together with our investigators, both Timothy Miller and Janice Robertson, to test their ideas and use this system as a bigger system and closer to the human system, and of course also very valuable to treat these dogs as well. So it's exciting to see the kind of tools we have available to us. I'll just remind you again, and it was already the opening comment I made, the confidence that we are going to be able to treat fALS when, I wish sooner than later. But the important thing for us in developing drugs is having the right target. And we debate long and hard what that target is, but there's certainly no debate for SOD1, C9. TDP43 and fALS are a little trickier in that they don't lend itself to just block it, like I'll show you we are doing with SOD and C9. Doesn't mean to say we won't solve the puzzle, we just have to be more creative about a treatment approach. Once we have a target, we go into the design of a treatment, and that in this case, we'll be talking a lot more about antisense oligonucleotides. That is being done to some extent in the academic sector, but I think through strong partnerships with academic and industry, we'll move this along much faster. The industry is just very set up to do drug discovery. And then, of course, we have to go and test into model systems. We can discuss whether it's the mouse model and which mouse model, perhaps manipulating the induced pluripotent stem cells, and I'll show you some data on that, is actually the way to go. So I'm very excited that as we move much more into the translational field, we are making stronger and stronger partnerships with the industry, and I don't believe that we can do it alone. The industry needs our network. They need you as the patients. They need the expertise of the scientists, and I think more and more we are integrating in much more constructive teams. So this is, of course, the excitement around the technology of Antisense, and you heard yesterday, both from myself and from Merit, that it's so exciting that ALS was really the forerunner of all this discovery. ALS was the first disease. I think through our ability to partner and fund a very risky idea in Don Cleveland's lab, driven at that time by a clinician, Richard Smith, and partnering with ISIS, that we were able to do this and really set the stage for all the other diseases that ISIS now tackles. As explained, the Antisense, if you are not familiar with the technology, is going to block, and it has to be administered not orally, but into the space around the fluid. What is very exciting about it is if you put it in that space, it actually travels and moves to where you need it. So we've had a lot of discussion yesterday also about stem cells. Where do you put those stem cells in? We're not so sure how far stem cells migrate when you put them in a particular place, and you saw how we had to get the surgical technique and put that into the physical tissue of the spinal cord. Here we're not going into the tissue of the spinal cord. We're just going into the fluid that bathes the cells, and then it actually moves. This was also quite remarkably a novel finding, because the company, their first disease areas were for retinal problems. And if you think about the eye, it's one focused area. The retina has a fascinating structure of cells and layers. And one example, macular degeneration, is a degeneration that happens in that region. And in fact, what you do is then put locally something. Here we are very lucky that, in fact, this antisense technology does move around the body as well as it does, and that was a novel finding, actually. So this then went into clinical trials, and as I explained yesterday, is going to go into new trials, and I think quite rapidly. ISIS is very committed to do this study. We just went back to the drawing board because they have better chemical moieties so that you can treat it longer term more safely. It was certainly absolutely safe for short-term treatment, but clearly, as you all understand, this is going to be a therapy that you will probably be taking early in the family as soon as we define what that time frame is, and for a long time. The good part about it, as I mentioned in the panel session yesterday, is that these antisense approaches, the molecules, they stay around for a long, long time. So the likelihood is you would only go for repeated but maybe monthly injections. And again, don't quote me on that time frame, but conceptually that's the idea. So then the C9 gene, and we'll hear a lot more about its discovery and from Dr. Trainer's perspectives where we're going. These are slides borrowed from my colleague John Ravitz, who is working in a team, and I will remind you, now that we've found the gene mutation and this expansion, we are funding many, many studies worldwide. Just yesterday we approved 14 new studies, 5 of which are all focused on C9. We also have obviously other priorities of funding SOD, FUS, and TDP43, but a lot is going into C9, and partly because many efforts in parallel are going to move this much faster. This is so unique because of this extremely large expansion. Might need a new pointer for the next session. But anyway, what you cannot miss is that yellow triangle, which is the expansion. And this was really very novel for ALS. Huntington's, myotonic dystrophy, all are called expansion diseases because they have these kinds of expansions. And this is just to quickly show how it was discovered and how you can see in a normal on the left. That's what it would look like if you're doing a test. On the right, there you see the expansions. And so this is from ISIS. They provided me with an update of where their project is going. We are funding them. Actually, they don't need any dollars as such because they are so well supported through all their programs. So they provide services in kind, but they want to be engaged in it and build the network again that we have provided and want to have the right people in the clinical trials. And so they are also partnering with Jeff Rothstein and the team there, and that's who we are funding. We are funding the academic partner. What they can demonstrate is that they can selectively target this region, so that's already been successful. They have shown that if you target that region in a mouse model, or I think it's rat or mouse, that's not a problem, because that's an important question, you see. If we are going to use the same approach as SOD1, is it going to be, we hope, safe, and is it going to destroy the normal function of that protein? We know for SOD that's not been an issue, but we don't know much about C9, this new protein, so we don't know if that will be an issue. And in this case, we are able to target it in such a way that we don't disrupt the production of the protein, whatever it's doing, but we do target the unwanted large expansion. And that, so far, in their study of a model system, is successful. And you have to remember, you know, the discovery of this was made in just almost a little over a year ago. For SOD in 1993, we spent years figuring out the problems, not even thinking about a therapy. We're thinking about a therapy at the same time as figuring out the biology. So what they are currently doing now is getting their best antisense oligonucleotide, the best chemistry, to optimize before they go into a clinical trial. But things are moving remarkably fast. So this is from John Ravitz and Clotilde, a team at UCSD that we fund. And again, in the first publication, it was described by Rosa Rademacher's team that what this expansion does is unclear, but what we do see that's abnormal are the red dots. And you shall see it much more clearly here if we highlight it here. And this is an accumulation of that expanded repeat that is abnormal. What is interesting and has just been published is that indeed, although this region, and we'll probably hear more about it in the next presentation, but this region is in an area that's not involved in the translation of a protein. However, what can happen now, because of this big chunk, is there is a way to start making something from it. And so although we see, first of all, the clumping of the RNA foci, what you also are seeing is a protein clump, which is going to be very interesting to define. This is shown also in the motor neurons, and these are actually from patients with carrying the C9. And what is interesting, because we are also so curious about whether this is typical for the motor neuron, or does it sit also in the cells around the motor neuron, because we've talked about the astrocytes being so important, and some double imaging studies have shown that indeed you do see these foci in the astrocytes. And so what they also have been measuring is to see how abundant are these. And this particular graph shows it's very variable. These are actually now in the skin cells from people that carry a C9 mutation. And they see that the number of cells that carry one of those inclusions is very variable from line to line. And if we see that the number per cell is also very variable. What all of that means, I'm telling you, very, very early data. It just gives you a flavor of the kind of things we're funding and the activity. We don't know what all of that means. What they are doing together with ISIS, while ISIS is developing the therapeutic, they are characterizing in fibroblasts what happens when you shut it down. And this is just showing you they're taking the antisense, they're blocking that C9 expansion, and so what you're seeing is the level of the RNA now in those that are untreated, of course, should be and is much higher. Those that is treated is much lower. So it's just telling you that that antisense is working to stop the production. And these are all done then in fibroblasts. And this is what is so interesting in this particular opportunity. You know, we don't have to rely as heavily on mouse models because we now know we can take skin cells with their genetics and we can go straight to human cells to test all these questions. So it gives us a little bit of an edge having both the technology of iPS, stem cells, and being able to go straight to the human patients. This is just not even for you to really digest all of it, but to say what they are now doing is looking at cells in which they have downregulated and blocked the production of C9 compared to cells where they have not, and are starting to look if we can find signatures. This is all to really look at biomarkers. Are there things that are going on that are different in the one and different in the other? And these are technologies that are really just showing and helping tease that out. This group, as well as Johns Hopkins group, are doing similar things, and I think aligning what they find will suddenly pool in and hone on a much narrower field of what's really interesting because we get so much data coming out of it. So in summary, one of the exciting things, and I'm sure that Dr. Trainer will emphasize it, is we have a gene that really links frontal temporal dementia. We have in the past over time known that there's been this combination, found interesting signatures that are similar, genes that are similar, but this is the first mutation that links frontal temporal dementia and ALS. It also gives the extremely interesting question of how does one mutation affect two very different systems, and so we obviously are interested in that biology. It's also the first expansion disorder, expansion repeat in ALS that we found, and that now puts us in a group of diseases like Huntington's, as I mentioned, and myotonic dystrophy. And of course, all of those investigators in the myotonic dystrophy field are applying to our programs because they have technologies that can help us with our problem. And so last night we approved the funding for 2 individual teams that have really been focused on myotonic dystrophy in the past. Well, the LOF is just a loss of function, so is the toxicity because we are losing some function, or is it because a gain of toxicity? And they were exactly the same questions we asked for SOD1. We quickly came to the conclusion in SOD1 mutations that it's not because you lose the normal function, but it's because you gain something new. And that was the rationale, therefore, for blocking, because we don't, whatever the new thing is that we might not all agree on, if we just block it, that would be an approach. And finally, it really talks to the RNA biology being so important, and I tried to show you some video images, which I'm not going to show again yesterday, of what's going on in the cell and the complexity of all of that, and now the fact that we have many genes, TDP43, FUS, and C9, all in a similar functional biology, quite distinct actually from what SOD1 is doing. So interesting that we have a different category now of genes that are involved in RNA biology. And that really it gives us the encouragement that ASOs are just antisense oligonucleotides are going to be a way forward for treatment. So this is really all credit to Clotilde, who was a postdoctoral fellow with us, who's now really an established investigator and has now got other exciting positions and moving on, but staying in the ALS field and done wonderful work. This is also, again, a strong partnership with Frank Bennett and his group at ISIS and many other teams at UCSD. And it gives you a very nice example. These are really all credit to John Ravitz, but the number of people involved in a study and the diversity of expertise, which is so important. They are supported not only by us, and it's really a team effort to do that work. So then switching gears, Dr. Benneter was very sorry that he couldn't be here, and we fund him also in many, many of his studies, and I know last year he updated about the study with the presymptomatic fALS study, and basically we fund him now to support work not only in the SOD, but also C9, FUS, and TDP43. And many of you are part of those studies. The goal of it, of course, is to understand much earlier what's going on in the disease, getting patterns from blood, from imaging studies, and this is just to tell you that obviously if you're not familiar with it, it's a visit to Miami every 12 to 18 months to do various analyses. And as I say, he's doing MRI. I showed you a little bit yesterday what EIM is. It's really a measurement of muscle strength. Muni is also a measurement of muscle activity, And in fact, combining this might give us a very good profile of what is happening. So just to give an update, he has 190 who have enrolled and consented. And really, as you know, you don't have to understand what your genetic background is. That just remains within the study. He has enrolled 73 into the program, and really, if you want to know any more about it, please contact him. If you don't get time to get the email, come and get it from me afterwards. But I think it's really important, you know, as we talked about yesterday, everyone would like to be in clinical trials. It might not be appropriate at that time to be in a trial, but everyone could be engaged in this, and it's such an important part of our research program. For those of you who have learned a bit more about the arimoclomol, this has been a very, very long study, and I'll just give my perspective and then share the data from Michael. When at the time we thought we should think about treating fALS in a separate way to how we think of the sporadic disease as a whole, because here we know the target, the SOD1, maybe we need to divide up in subsets. And we talked a lot about the challenges yesterday that clumping everyone together with such a variable disease, maybe there are responders for drugs and non-responders. And so with our program that we had initiated to be able to fund novel and exciting ideas, Michael was one of the first actually that applied to us. And we worked together as a team to think about it. If we were to then think, you know, only including the SOD families, what kind of drug would we take to test? And of course, our portfolio of compounds at that time was minocycline, which later we found was toxic, a variety of things. Why arimoclomol? Well, at that time, there was some data to say that this was a compound which actually had already been developed. This company, Citrix, bought this compound, and it is a different enantiomer to the one that was established by arimoclomol. What it really worked on was a concept that SOD misfolds, and somehow it works on the misfolding. So we thought, well, this compound at least is attacking a very specific mechanism that we think is going awry with SOD1. It's just a challenging story because, number one, you get very few, there are many families, but of course, over time, we lose families to SOD1. And to enroll in a clinical trial, especially if you're looking for a very small effect, you need a lot of people in that trial. So to get the number of people, to keep the commitment from the company, who the company no longer is focused on ALS, but we were able to ensure in the contract that we set up that we would have access to the drug. So this has been a very slow and long procedure. I think what is most important to me is it sets the stage for how we're going to do this in the future. What have been the challenges? How can we enroll better? And I think many lessons have been learned and how to set it up. Whether arimoclomol is the best drug is another question. It may have very little effect, but it sets the stage for doing this as we get better drugs. And one other thing that was important to do, and Merit talked about it yesterday, if you do phase 1, then phase 2, then phase 3, there is an advantage to that, but the challenge of it is then you take phase 1, have to analyze the data, get approval for the next phase, do phase 2, makes for a very long study. This is an adaptive design where phase 2 and 3 will go straight from one into the other. So in other words, there is an oversight committee, it's called a data safety monitoring board. They will know the data, it remains blinded, so they will know if there are concerns and safety issues, and they will tell the investigators to stop, but the investigators don't know what the results are, what's going on. And then at the phase 2, there is a discussion with those who are allowed to see the data, whether there is good rationale to move on, but whether there seems to be some hope that it's going to work, and then it goes straight on to phase 3. That also allows you to put the same people into the phase 3 design because they haven't been blinded or unblinded. That means for a trial where you need small numbers, that's huge value. You can move along into the phase 3. So this was really a design that hadn't been used much before. Ceftriaxone, and Merit talked about that, was the first, I think, in ALS. And this is another design that really borrowed from ceftriaxone's lessons. Again, here he highlights phase 2 is the safety, and phase 3 is the efficacy. The more people you have, the more likely you're going to see an effect. So it just grows in size. It's randomized, placebo-controlled, etc. There are 2 sites enrolling, Miami and MGH, in this trial. And this is for the SOD1. Particularly, I don't think there would be any value at this point to bring in C9 because we don't think that that's the biology that's happening. So it's specifically because of the SOD1 biology. The current status is that 74 people have been screened and 34 enrolled that are eligible for the study. So small numbers, that's really the challenge. And we'll have data soon. And then he too is working on the Antisense project. I'm going to go quickly through it. I don't actually have a clock with me, so someone will have to wave when it's my time up. But really, this is why it's so exciting. There really are 3 groups that we fund with slightly different techniques to do the Antisense technology. And I won't go through too much more of that because it's similar in concept. They have optimized and set up systems. They're really designing in a very different way these antisense oligonucleotides. They might work better. They might have similar effects, but approaching it slightly differently. I will just quickly go through this. And then I wanted to highlight this study that's going on with Martin Turner in the UK called Biomarker. And he too, in fact, they work closely together, Martin and Michael, and there's actually a consortium around neuroimaging so that they learn from each other how best to advance the imaging technologies. But Biomarker has a similar principle that, in fact, if you do both the imaging, the blood samples, the CSF, you'll get a combination of signals that together will tell you a lot about the disease. And I showed this yesterday. This is from Martin's group. He has developed with others, many others, Eric Puro and others, technologies that rather than looking at post-mortem tissue, which this is to see where things are dying, he can look in live time to see how things are changing. And what has been the challenging part of imaging really over time has been that, yes, we do see things are wrong. We see things are wrong very early, and they pretty much stay looking like that. So what we really need is to see a change over time, because then if there's a change, you can stop a change. That change is not significant enough to measure how effective a drug is. So imaging has been fairly limiting for that reason, because we can't say, imaging was the breakthrough for multiple sclerosis. The fact that companies have found treatments for MS is because they could image, see a lesion, and see that lesion change. That's what we want to be able to achieve, see that it's wrong and see that lesion change. We're not quite there yet, but we are using enormously advanced technologies that maybe that will come about over time. And maybe it's not going to be imaging for us. Maybe it's going to be something in the fluids or changes in small molecules. But this is just a clutch, and actually it's not so easy to see, but what he's doing is combining multiple measures, and all you need to know about it is that the green lines are the healthy, and they cluster in a group, and those that are ALS are in a different cluster. So without knowing who's who and then doing all these measures and then coming together and analyzing it, you see a separation in clusters. And that is the important thing, is to identify things that are different. And then just to remind you that I think being supportive and giving infrastructure support to the network for clinical trials is not only valuable for clinical trials that are ongoing at the moment, but also for research studies, and in fact the arimoclomol study, as well as the getting people involved in the SOD and the C9 trials, all are facilitated by these networks. And then just to remind you, for updates, we don't always have specific focus on genetics and the SOD and C9, but we always do have a monthly update on exciting new technologies or new studies. So welcome you to join our webinars if you're not already doing so. And with that,